Welcome to episode 1 of Life of the Past, and this has to do with life. And so life is that precious thing that we all hold, but at the same time there's a lot of controversy about what does it mean to be alive. And so we're going to delve into that a little bit today. There's extremes that are out there. So we're going to ask the question, first of all, what is life? What are the requirements for life? We're going to do a deep dive in Bermuda, and then we're going to talk about the Deep Carbon Observatory, and that is related to the video that you've been assigned to watch, Life's Rocky Start. And then we're going to talk a little bit more about extremophiles and endoliths. So that's what's in order for today. Before we get going, however, I need to give you a couple of announcements as well. We have a pre-course assessment, which will be available tomorrow between noon and 10 p.m. And so you will be able to take that pre-course assessment. That's when it's going to be offered, and it'll be 5 points extra credit if you take that. I think there's only 10 questions on it. So that's coming up tomorrow. So I want you to be able to do that tomorrow. If you look in the provisional course schedule, you'll see it listed as that time period for Friday, noon to 10 p.m. Other news to relate to you is the course textbook is ordered, but it's on back order right now, and so it should be available within the next day or two. They're expecting that shipment in to the Missouri State University bookstore. So look for that if you're over in that direction on campus. So without further ado, let's dive into life here. So, okay, just to explain a little bit on the title slide is Life, and Life used to be a magazine. When I was a child growing up, my parents used to read Life, and so Life was famous for its photographs. And so on this photograph here, actually it's not a photograph, it is a artist illustration for what it looks like to be an anglerfish. And so we'll talk about those sort of extreme settings in which animals live on this planet in this episode. So what is life? There's no consensus. There are things that are alive and things that are dead. And we say that all the time, right? So it was once alive, now it's dead. But there are things that aren't living at all. But how do you go about separating those two out? So there are certain characteristics we expect out of living things. However, modern technologies kind of blur the lines on this because we have artificial intelligence now. We have self-driving cars. We have robots that will be able to think and make decisions. And then also there are things that we're part of living tissues, but we can put into that some sort of capacity to heal and things like that. That's the xenobots, for instance. Xenobots are sort of some type of frog tissue, I think it is, but they've been designed to actually repair themselves. And so what's living? Oh, and you and I know over the last 2 years we've had a pandemic. Are viruses living? Some people say no, because they are not something that just is in the natural environment and would be able to reproduce and metabolize and do all those sort of things that we would expect of a living organism. But in fact, other folks make the argument, in fact, that viruses are living things. And so that is because they can get into living cells and then totally corrupt that cell into making more viruses. So it benefits the virus to be in that host, and that host can then be hijacked essentially to make more viruses. So viruses like it's the coronavirus, SARS something or other, SARS-2 I think it is, virus is it's not an organism even, right? It's a structure, it's a protein essentially wrapped around this sort of RNA structure. And so RNA, ribonucleic acid, and it allows it, once it's in a host, to be able to replicate in great quantities and then spread to other host organisms. So some people think that viruses are alive, and I think you and I probably would fall into that category now, saying, oh, yeah, they must be alive because they evolve and they get these different variants and so forth. So when we describe living things, it's usually not cut and dried. And so what we have instead are characteristics that we use to describe them. They tend to be open systems. They have homeostasis. They require energy in order to live. homeostasis, self-regulation. They're composed of cells when we talk about things above the virus sort of level of organization. They have a life cycle, so they begin and they, mature and then eventually die, right? And so that metabolism is the energy conversion that they do in order to survive. And the actions that they do, they grow, they adapt, and they usually respond to stimuli. And then lastly, they can also reproduce, right? So you have to be able to replicate in order to maintain that living structure. And they evolve through time. So with that in mind, if we just describe the characteristics and, not say that there's anything that's actually hard and fast between is it alive or is it not, I think we're probably safer that way. All of life that we know, on this planet anyway, has RNA or DNA. So deoxyribonucleic acid or ribonucleic acids. And so those acids, in fact, are composed of a few essential elements. And so the elements for life are in this order. And this is probably going to be on the first quiz, which is next week. That is carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And even some of that we can kind of stretch the boundaries on slightly. In 2010, there were NASA scientists who were working in a place called Mono Lake in California when they discovered that there seems to be a bacterium there that could actually take arsenic and substitute it for phosphorus. And so the phosphorus, in fact, is a very good regulatory sort of element for our bodies. But in some cases, arsenic can actually, which is a poison, arsenic is a poison, it can actually replace the phosphorus that has a similar structure. And arsenic is usually pretty bad for most living organisms, so it has to be a pretty extreme environment for people to say, oh, I want to use this, arsenic, instead of phosphorus. And in fact, only recently they've discovered that, well, it's not that recent, I guess, but they've discovered that it will only do that when it has to, okay? So when phosphorus is in such low supply that you actually can then substitute the arsenic to take the place of the phosphorus, and not all of them do it. Just one bacterium that does that. So people speculated because of that, maybe there's a higher probability of having life on other planets if you could use arsenic. Because phosphorus isn't always available in most planets. And also recently they've discovered the gas, I think it is, phosphine, but it's very trace amounts, okay, in the atmosphere of the planet Venus, okay? So there's been some speculation that maybe there could be life on Venus, but it's not really likely, okay? Venus gets a lot of energy from the sun, of course, and you would have to be up in the atmosphere, in other words, to be living in Venus, because the conditions on the surface of Venus are too far beyond the type of conditions that are conducive for life, at least on this planet. Okay, so we speculate that probably Venus doesn't have life because it's equivalent to, if you know geology at all, it's equivalent to Greenland facies on planet. So it would be deep inside of the Earth, essentially, where those sort of things could happen, where life could occur on a planet like Venus, under too much pressure likely, and probably too hot too. So with that in mind, those are the 6 elements that go into life as we know it on this planet. We don't know of life on any other planet yet. We're looking for it, but we haven't found it yet. We're looking. Just to let you know, we're looking for stromatolites. We're gonna talk about that. It's actually in the PBS video that you watched as well from NOVA, and they're looking for stromatolites on Mars. And so, but that's been a long time coming, and they haven't found it yet. So let's go on with life on Earth, okay? So with that in mind, what is life? What are the essential components of life? What are the characteristics of life? Let's go on a deep sea cruise. The New York Zoological Society sponsored a man named William Beebe to do some research on the island of Bermuda. Now, the island of Bermuda is in the Atlantic Ocean, and some people think that it's actually regarded as a hotspot. Now, a hotspot is one of these places on Earth where there's essentially a volcanic sort of origin to an island, And in this case, that island would have risen off of the sea floor and been up at altitude or in the bathymetry for a certain length of time, and then carbonates would form over that if the conditions were correct. That's still up in the air if it's really a hot spot or not. But we do know it's a very isolated island, and it has very steep sides on it. And so William Beebe decided, is like, well, let's build an apparatus to find out what kind of life lives half a mile below the sea surface. Okay, it's below sea level. And he took on that expedition for the New York Zoological Society. So here, in this next image here, you can see a satellite image of Bermuda. And it's actually, north is up to the upper right here. Okay, so it's not north and south. I had to get it across here just to show you this. But William Beebe proposed going to one of these areas where it's steepest, and that's on the island of Nonsuch. And so off the island of Nonsuch, actually, they decided to lower an apparatus. Now, Bermuda has a long, long history, okay? So it was first found by the Spanish, who passed by it, essentially. And then later, it was colonized by the British because they needed a place where they could stock up on water, fresh water to drink for the sailors. And then also they would be able to grow limes here. And so limes would then keep the crew safe and not experiencing the deleterious effects of, let's say, scurvy. You know, scurvy is one of these things that affects sailors because they're at sea for too long. They have a deficiency in vitamin C. So vitamin C is one of these things that's essential for life, in humans anyway. Here's the island of Bermuda from way back in the 1800s here. Actually, 1692 here, this map here. That's one of the earliest maps of Bermuda. Here you can see, in fact, from a book called Half a Mile Below the Sea, I think it was. This is the island of Bermuda. You can see where the coral reefs kind of surround the island here, but they sent the bathysphere, or the bathyscape, off to the side, or the bathysphere off to the side of the island of Nonsuch. It was the New York Zoological Society that proposed this. William Beebe was that man. He didn't know how to do it quite, and he proposed building a cylinder, essentially, to lower a human being in a cylinder that is supplied with oxygen and communication means all the way down into the seafloor, half a mile down. Well, there was a young man named Barton, William Barton. Oh, no, it's Otis Barton. And Otis Barton said, that's not going to work because the pressures are too great at half a mile down, that would crush whoever's in there. And so he says the only way to do that is to withstand that pressure is to build a circular or a spherical sort of structure out of cast materials, so cast steel, if you will. And so what they did is Barton designed this sort of bathysphere to withstand that pressure. There were a couple of windows and a hatch on it, so you could look out and record what you saw and have some lights that project onto the outside as well, and then have communication up to the surface ship that was allowing this thing to be tethered, okay, so they would be able to retrieve it on a wire rope, essentially, up and down from a British ship. So the folks that were involved in this was an experiment, by the way. It was before World War II, in the 1930s, so in the 1930s, and they went to Nonsuch Island. William Beebe did not have credentials, as it turns out. He was not a doctor, he wasn't a professor, but he was an ornithologist, so he was a bird expert, but he had that idea. And so they said, well, we'll fund it, so we're going to let this happen. Otis Barton was an engineer and a wealthy young man, and so he actually funded part of the structure, the building of the bathysphere as well. He designed it, he engineered it, he built it, and I think he kind of felt like he was left behind in William Beebe's attempt here, to gain notoriety. Well, William Beebe wrote a few books. In fact, I have one in my library here, and if you'll pardon me a minute, I'll just go show it to you here. This is the book he wrote, Nonsuch. This is one of the books, anyway. The other one was called A Half Mile Down in the Oceans, I think, something like that, anyway. But anyway, it was quite an adventure. So In this photograph, you can actually see William Beebe at the top. You see a line drawing of the bathysphere. And now below this, the only person who really had credentials in this whole expedition was the woman who was hired by William Beebe to be the communications person. And so that's Gloria Hollister. She's actually an ichthyologist. She studies fish. And she is, as Barton and Beebe are, they're all explorers. And so we're going to go into this deep place inside of the oceans to see what happened. Here's some photos from that book. And you can see here the hatch with William Beebe staring out of it there, and then Barton and Beebe below that, and crawling in and out of the bathysphere here. It's a very small structure. In fact, this thing was almost lost. The bathysphere was stored away, and then finally they pulled it out and say, oh, well, look at this, and so we found this old story. And so they've since rebuilt it essentially to show at museums and so forth. Here they are lowering the wire rope, and you can see it's tethered by that wire rope and some communications cables, so it could be lowered into the ocean. They had to test it many times first, right? So when you test these sorts of experimental craft like this, I mean, it's like shooting the moon, shooting a moon project here. It's the first time that anybody had ever been to that water depth before. It's deeper than any of the submarines at that time could tolerate, and certainly deeper than the ones from the 1960s. All of that's been surpassed now, but for now, anyway, this was really modern technology. When they were testing the bathysphere, and they lowered it to a certain depth, and they brought it back up. One time when they were removing the bolts from that, it was under such pressure. I think there must have been some sort of pressure in buildup inside of the structure that they couldn't bleed off properly. And they, as they were undoing the bolts on that, one of the nuts from the bolt shot off, and so it was kind of a risky sort of business. But they risked their lives in order to explore and understand more about the deep sea. And so here they are standing next to the bathysphere here. Here it's being lowered over the side. So after all of the trials and the British ship that was there to actually see that the experiment was carried out, because at the time, and still now actually, Bermuda is a part of a royal colony. Right? It's a royal it's a British thing, right? So it's the United Kingdom who actually owns Bermuda. They lower it down and they begin taking photographs. And here's one of the images of one of the early fishes that they saw down there. And of course, if you were to just fish and try to bring these fish back up to the surface, they would be under so little pressure that they would actually explode on the ride up. In fact, I've heard about people, even in the Caribbean, right? If you get several hundred feet and you have a fish trap deep inside of the Caribbean, and you pull that fish trap up, it is going to inflate all of those fish because they all have air bladders in them. And so most of the fish become distorted, but they still eat it, right? In this case, these were just there to see. And so it's a way to see the fish in their natural environment. So that's kind of a more non-destructive sort of technique. And they saw many things that have never been replicated. So some things that William Beebe described from at depth, and he was talking over a microphone to Gloria Hollister at the surface and would describe the things that he saw and then sketch them. As he sketched these things, they had inside of here, of course, they had air inside of there that was in compressed tanks, just like scuba almost. And then they also had a sort of recirculator so they didn't oversupply that cabin with carbon dioxide because that would knock them out first and then kill them. So they had a scrubber that would actually remove the carbon dioxide from their exhalations. He would draw things, and some of those have never been replicated. We don't know for sure if he saw everything that he had sketched or whether those sketches were actually accurate. Okay, so, but even so, it was an exploration in things that people had never seen before. Things like the anglerfish that you saw on the first slide there. Okay, that was in the 1930s, 1930 to 1934. In 1939, Nazi Germany invaded Poland, and for the next 6 years, the world was at World War II. And so after World War II, 15 years after World War II, they decided we need to go back and replicate these sort of experiments that William Beebe did in the bathysphere. And so they came up with apparatuses like these two right here. Now that's the Trieste on the left-hand side, and that's the Alvin on the right-hand side, but they were deep-sea submersibles that were actually manned vehicles for people to get into. And so in the Trieste here, you can actually see that is a bladder at the very top of that. So that's actually only a tank, and the people would be down in that little ball, still no bigger than what the Bathysphere was, making observations down there. There's one other type of submersible that they use. It was called the Bathysphere, or the Bathyscape. So the Bathyscape looked a lot like the Trieste. Okay, so the Trieste, I think, was a design improvement over the Bathyscape. Here they are lowering it, and so these were additional sort of discoveries that were being made in the 1960s. And National Geographic and some other organizations helped fund many of these expeditions. So what did they find? They saw fish like this. Now, occasionally you'll actually see an anglerfish that gets washed up onto the beaches of the Atlantic. But on the left-hand side, you can actually see a model for what these things look like. And that's an anglerfish that has a little light at the top, which would attract prey. And, of course, it would be able to then bite and eat whatever was in reach, I guess, for that organism. There are other deep-sea organisms that I have very little understanding about at all, but I think it's some sort of cod. But here you can see what struck people is when they first saw this organism, this fish brought up from the sea, they were struck by the fact that it had something like a human face almost, and so that is an actual photograph of a sort of fish that would live at great depth inside of the ocean. The next image is just there pretty much for shock value. Notice that the eyes are really large, and I assume that's to withstand the pressure so it wouldn't crush the retina in these fishes. But again, you're under really low light conditions, and you have to have large eyes to be able to make out anything with vision, or you have to have some illumination sort of device to be able to see what you're around. And I think these, I don't recall what the name of those are. Expeditions kept going into the oceans, not into the deep oceans necessarily, but into some of the colder oceans. And so, for instance, in the 1990s, I think it was, they found this organism in the oceans around Greenland. And so in Greenland, this is a Greenland shark. And the fascinating fact is that the Greenland shark is one of the oldest living animals on the planet. So I think that when they caught one of these and determined its age, it was around 400 years old. That's pretty old for a shark, right? That's older than what human beings live by a factor of 4. Deep sea submersibles kept being sent down, and then eventually they became unmanned exploration devices. And so in the next image, these are the sort of tubeworms that you get around places that are deep sea vents. And so there are communities that are developed in places where there's, well, some hydrothermal activity around volcanoes and places like that, and also around subduction zones, and in any place, in fact, where there are vents, essentially, that would have an exchange with seawater and water that is inside the upper part of the ocean crust. And so these things are tubeworms. And I recall when they first discovered the tubeworms, and I think some of these got up to like 6 or 7 feet tall. And they were usually around something they were called black smokers or white smokers, and I think both of those, in fact, would emit minerals into the water. The black smokers had a lot of iron and sulfur in there, and so the white smokers, in fact, had some other chemicals in there as well. And so these are worms that developed. And what you're probably wondering is, how can a worm live at great depth like that? What do they eat? They have to filter feed. And in fact, they usually feed off of bacteria. So bacteria is one of the key things. And in fact, I'm in the process right now of studying an area where there seems to be a methane seep, even in Missouri here. Okay, you wouldn't think that. But geology is a funny business. Okay, so in the deep water deposits of southwestern Missouri, there are some chert beds where there are worms that were preserved, and they are preserved in chert. And in fact, so what do you eat when you're in there? Well, most people would speculate that these things, the worms themselves, would actually eat bacteria, and they secreted tubes on the outside to protect them from the forces of high pressure, and then also from predation and things like that from the crabs and other things that would live in that area. So that's around a vent community here. So, in fact, we're really talking about places that have extreme sorts of settings, right? So hot water is one of those. There are other places as well. And in fact, one of the things you're going to get to know a little bit about me because I'm going to tell you stories all the time, okay? So, in fact, one of those stories revolves around going to Antarctica. I went to Antarctica in 1989 for the first time. I was there from 89, 90, 91, 92, and then 92, 93. I went there 3 years, went there for like 3 months each time during the summer months in Antarctica. When it's just barely below freezing, well, okay, we got down to 55 degrees below zero at South Pole when I was there, but other than that, most of the time it was around 15 to 20 degrees. I did get my nose nipped one time by frostbite, but it came back okay, so I didn't lose any tissue out of it. I was in Antarctica collecting mostly limestone samples. We were looking at some rocks called the Nelson limestone. We also looked at another rock unit that's exposed in the Argentina range. So we were up on we had to fly to Pole just to get to the mountain ranges that we were studying on the Weddell Sea side of Antarctica. And if you don't know, Antarctica is the coldest, highest, driest continent on the Earth. You wouldn't think that it would be the driest because it has so much snow and ice covering it, but in fact it is a desert. It's a cold desert, and it sets up at relatively high elevation. And so in that sort of setting, when I would sample these rocks, a party of 4 of us were out in the field, and we do field geology, we would sample pieces of limestone. And when we were ready to be picked up by the C-130s that were ski equipped and they would land on the ice, pick us up with our gear and the rocks that we'd collect, all the samples and everything, right? We would wrap those samples in plastic and duct tape them and then pack them in boxes very carefully so we didn't crush anything. And so they would make it back to our labs back in the United States. I was at the University of Kansas at the time as a graduate student. And so when we loaded those boxes and the C-130 would pick them up and take them back to McMurdo Station for us, they would have to wait until the supply ship would come in, deliver the food and other supplies to the McMurdo Station, all the other field stations in Antarctica, and then they would take the rock boxes back with them. And so those rock boxes went from Antarctica, from McMurdo Station, up to Port Hueneme in California. It's on coastal California, a little bit north of Los Angeles. And so at Port Hueneme, they would unload these things, palletize them, and then send it off. And so it was about 6 or 8 months later that we got these boxes shipped to us by the National Science Foundation. I was responsible for like opening these packages up, right? And so you would cut the plastic off and take a look at the rock. And then sometimes we would look for fossils inside the rock. So you would take and crush the rock with a splitter, essentially, so it would break the rocks for us conveniently. When you split those rocks, sometimes, quite often, you would see a green rind around the inside of that rock. Now, this is not a rock from Antarctica, I don't think. It's not one of the rocks that I collected, but this is an image that I grabbed off of Wikipedia, I think it is, but it's an endolithic algae. So endolithic algae live in the harshest environment, harshest subaerial environment on Earth, that is, not below the oceans, and they would form these sort of like layers of living organisms inside of a rock. That's pretty tough, okay, for an algae to live in Antarctica and try to wait out the winter there and still reproduce on just below the surface where it gets just enough solar gain to be able to, well, when it finally got exposed to warmer temperatures and to the sunlight, it could photosynthesize, actually. We call those extremophiles. And in this case, this is an extremophile that is an endolithic algae. Endolithic algae, in other words, inside of a rock, endolithic. And so endolithic algae are one of those sort of organisms that lives in extreme conditions. If we go on and talk about other things, now I did already mention about the arsenic metabolism that certain organisms have, bacteria in Moto Lake. Well, this is in Moto Lake here, and these are what they call tufa towers. And so tufa towers are sort of structures that algae can actually make when they're submerged. Well, these are high and dry because they've drained the lake, but they still live around the perimeter of these sort of tufa towers. And so those algae that are in the lake waters surrounding these little islands of structures that were associated with extremophiles, those are the ones that actually could metabolize arsenic. But of course, as I just already mentioned to you, they substituted for phosphorus, but only when they had to, okay? So there are some that, well, they're able to metabolize arsenic instead of phosphorus. There's a lot of different extreme environments that organisms can live in. One of those, if you've ever been to Yellowstone National Park, you would know that many of the hot springs there actually have colors associated with them. And in fact, here you can see some green ones. Well, the green ones tend to be the lowest temperature. Things that would photosynthesize would be able to live in the cooler temperatures, but the yellow temperatures would be things that perhaps are sulfur-fixing bacteria. And in fact, many of these things may not even be algae. They may actually be cyanobacteria, or, as we say, cryptomicrobial sorts of organisms. That is, in other words, they have the ability, archaea, that's what I'm looking for, archaea, that actually photosynthesize. And so this is a hot spring, in fact, on the left-hand side in Yellowstone National Park. So life can find these extreme sort of conditions and still survive. They can survive in rocks. They can survive in hot springs. They can survive in salt lakes, essentially. And so on the right-hand side, this is an area that only gets wet during heavy rains. And you don't get very heavy rains very commonly in Death Valley, but this is essentially a salt crust. And that salt crust is actually host to a number of bacteria and archaea. Archaea and bacteria are kind of like this. Bacteria are a little bit more evolved than the archaea. So the archaea are some of the more primitive sorts of single-celled organisms that are able to thrive in extremophile conditions. So if we took the sum of all the extremophile conditions that you could see on Earth, here's kind of the panoply of these sorts of conditions. You can get them in ice. Ice can actually change colors. There's raspberry ice, I think it is, that has a sort of reddish microbial sort of growth in it. Cold water seeps, mud volcanoes are another sort of setting. Hydrothermal vents we kind of talked about, but they can either be shallow or deep water. Hot springs, obviously. Even volcanoes can have, volcanic lakes can host, because they have the right minerals to be able to metabolize and to be able to do all the things that living organisms do. And then deserts, of course. Deserts, even on those top and bottom surfaces of deserts, when it gets cool, water can condense on rocks, and so you can actually form what they call desert varnish in deserts. And that's actually because of magnesium and manganese fixing bacteria. That black coating that you see on some rocks in deserts, that's the result of living organisms. And then lastly, even in mines where we mine out coal or we mine out minerals that are like metallic minerals or semi-precious minerals and things like that, even from the waste deposits from that, algae and cyanobacteria, archaea and bacteria, they can live in those sort of conditions. That's an extreme environment. So we call these things extremophiles. Endolithics are just one variety. There's all of these other varieties that can exist. So deep sea anoxic, in the deepest of the oceans, we still find living organisms. Hydrothermal vents. Serpentine is a type of rock that forms in the ocean crust. And even in serpentine, they find living organisms, deep sea sediments, of course, in the trenches. Continental margins, of course, are going to have a lot of that, and you'll find fossils associated with it. Even in nuclear contaminated sites, you will find biological organisms that will actually uptake some of the waste products that are there. And so it's really kind of wild life that we have on this planet. If you can imagine an environment There's probably a living organism there. And that's why we think that even on planets like Mars, we potentially may be able to detect life at some point, because we know that there's water buried there. We know that there's carbon dioxide. Well, we know that there is a very, very minor atmosphere as well there, but they do get solar energy, and it was once covered by water, so why wouldn't you get things like some of the primitive things that we see on Earth, the first living organisms that we find in fossils, like stromatolites, some of the most abundant of the macrofossils, let's say, that represent some sort of living organism. Those things are still present. And so, so I asked you to watch the video, Life's Rocky Start. That is a documentary that is, well, it's produced by PBS and the Nova series, and it's all about Robert Hazen. And so Robert Hazen's this guy over here. I think he's at Harvard, but he talked about the different colors of this planet, essentially, and how life and Earth are intertwined completely. If we had a dark start made out of molten material that eventually would crystallize and form a rock, that material would become weathered in the environment that was produced by the volcanic gases that were emitted by volcanoes. And after that, you would be able to have cometary impacts on this planet and producing some of the earliest oceans. And some of that water probably came from the mantle and was released from the mantle as well as juvenile water on the planet. And all of that produced, it allowed stromatolites to form as early as 3.5 billion years ago. After all of that, we finally developed, through photosynthesis and through the actions of stromatolites, an atmosphere that would produce oxygen, and we wound up with what Robert Hazen called red Earth phase. And then after that, because we had an atmosphere and because we were able to produce carbon dioxide, all of a sudden we also developed the first ice ages. So there was climate change even in the earliest Earth. And that almost wiped out life as we know it. If it wasn't for the volcanoes, life probably would not exist on this planet. But even after the ice ages of the Ediacaran or the Cryogenian, there were a couple of major glaciations early in Earth, and so that would have left what they call a snowball Earth sort of hypothesis. It would have left a very cold, sterile planet if we'd have only had that. But instead, the volcanoes warmed things up and life returned to Earth, and we've had a living planet with unusually complex cellular animals for the last 600, 700 million years. And so that's kind of the story. And that's where we start when we talk about life on Earth. And so those are the 6 colors of Earth that are described in the video that you're assigned to watch. And I guess that's it for now. We've talked about life. What is life? What are the characteristics of life? What are the essential elements of life? That's important. And remember that all living things that we know of anyway have RNA or DNA, so they have to have those sort of structures, those chemical structures, in order to be able to survive and live and have a life. So, well, here we are at the end of the first lecture. Thanks for your attention, and I'll come back later with talking about time. Because I've just talked about these 6 stages here, you have to have a little bit of background and understanding about time as well. So we're going to leave it at that. If you have questions, email me. That's the best way to communicate with me. And remember, you've got a pre-course assessment tomorrow. It's not a quiz, but it is worth 5 points of extra credit. Okay, so please take that if you would. I'd appreciate your cooperation with it. And I know this is coming at you late. I just got back from Jamaica 2 days ago to talk about a place that has a lot of life and teeming with life. What a great place. Anyway, thanks for your attention. And, well, cheers. I'll see you in the next episode.